We have just seen how so much of the accessory organ function is to contribute various substances to the digestive tract, and specifically empties it into the lumen of the duodenum, the first part of the small intestine. Let's look at the big picture of all of those processes, and then we'll look at the small intestine itself. This illustration demonstrates the importance of our signal molecules cholecystokinin and secretin, and how being released from the mucosa of the small intestine, specifically the duodenum, into the bloodstream, they then have a variety of targets. They target the liver, they target the stomach, they target the gallbladder, and they target the pancreas. Why? What causes them to be stimulated? The contents in the lumen. If the chyme is high acid, which means low pH, high fat, high protein, that is going to elicit the secretion of these two signal molecules, these gastrohormones. I want you to look through this step by step and make sure that what I have told you in the previous parts of Chapter 1, that you understand these mechanisms. If not, let's talk about them in class. All right, now let's go on to the small intestine. We've talked a lot about the duodenum, but let's take a look at the fact that it has 3 sections. The duodenum is the shortest and is just this kind of curved part. The next section is the jejunum. A lot of digestion and absorption happens there. And then the last part is the ileum. Please note the spelling. Ileum with an E is the small intestine, where ileum with an I is part of the pelvis. At the end of the ileum, there is a connection, a regulatory valve, that allows the chyme that is going to leave the small intestine to enter the large intestine. And we'll take a look at that mechanism later. But this piece of the large intestine is called the cecum, which is why this is called the ileocecal valve. As I often say, how it's built dictates how it works, and that is certainly true of the small intestine. So let's take a look at the histology of it rather closely. The first thing to look at will be the mucosa. Of course, it is covered with epithelium, but this epithelium is really, really cool. Now, the epithelium is made of simple columnar epithelium, but it has the modified surface on the apical surface of the cell that we call microvilli. You are going to have to reach back into A&PII about that. Increased surface area from the microvilli increases the amount of absorption that can happen here. So the microvilli by themselves really increase the rate of absorption. These cells cover what are called villi. One villus, many villi. The villi project up and increase the surface area. So there is another level of increased surface area. And these villi are covering additional extra folding of the lining of the small intestine that are called plicae circularis, or circular folds, as you see here. So we have 3 levels of increased surface area, which makes the small intestine ideal for absorption. One more item to point out here on the mucosa. The microvilli is often referred to as the brush border, probably because it looked to somebody like a hairbrush. All of those microvilli projecting up towards the lumen of the small intestine. Let's take one more look at these structures, looking at the photomicrograph that we have here. This is a villus. It is covered with simple columnar epithelial cells, many of which are called enterocytes, and they are the cells across which your nutrient molecules will be absorbed. They will be absorbed into the bloodstream. We have a capillary bed within each villus. They also, fats, will be absorbed into the lymphatic pathway. And so we have a one-way lymphatic capillary called a lacteal. So those are also here. And then finally, structurally, we also see what is referred to as an intestinal crypt. So the villi project up off of the surface of the endothelium. The intestinal crypts are invaginations down from there. The intestinal crypts have unique secretory cells that we'll talk about next. You can see an intestinal crypt right here in the photomicrograph. So let's now talk about the key functions of the small intestine. Although we do not see the muscularis externa layer in this illustration, it would be deep to the submucosa. The muscularis externa of the large intestine engages in segmentation and a fairly slow peristalsis. And what this allows for is the chyme that is in the lumen is going to be mixed up by the segmentation, and that means it's going to get splashed up against the microvilli to be exposed to more enzymes there. In addition to splashing against the microvilli, the segmentation mixes the chyme up with the secretions from the pancreas and the gallbladder. So the enzymes come from the pancreas and the bile comes from the gallbladder. And that also is going to increase the third function, which is digestion. Here we see the final chemical digestion of carbohydrates, proteins, and fats. So we say that the small intestine is the final place where chemical digestion occurs that is triggered by our bodies. Don't forget that there are microbiota in the large intestine that can chemically alter substances in the chyme. But even though they're in our body, they're not of our body. Now, I've really made a point of our 3 levels of surface area increase, which leads us to the last function of the small intestine. Nearly all absorption occurs in the small intestine. I think about the large intestine as being where the final balancing act is going to happen in terms of how much water and ions are absorbed from the chyme. Our nutrient molecules are predominantly absorbed in the small intestine. This is a really wonderful electron microscope image of a surface of one of these enterocytes or absorptive cells that line the small intestine. So this is one of those cells here, okay, and this is a modified surface area projection called the microvilli, and they form the brush border. Now, on the surface of the microvilli, we find intestinal enzymes. They are not secreted into the intestine. They remain on the surface of the microvilli, which is another reason why segmentation is so important, to splash the chyme against the surface area of the brush border so that the enzymes that are part of the brush border can target what essentially at this point will be either dipeptides or disaccharides or very well emulsified fats. This is a really good table that just simplifies the control of small intestine motility. In other words, how would we speed it up and how would we slow it down? Well, if you increase gastric motility and emptying into the small intestine, that increases motility in the ileum and relaxes the ileocecal valve. In other words, you put more in from the stomach into the duodenum at the beginning of the small intestine. At the tail end of the small intestine, you will be moving more substances out of that part of the small intestine and into the large intestine. This is referred to as the gastroileal reflex. It is a long reflex. Gastrin can trigger the same thing. On the other hand, we may see distension of the small intestine, and that will increase the strength of segmentation, and that is triggered by both long and short neural reflexes. The flip side of that is to reduce intestinal volume. For example, if you're fasting, this initiates what is called the migrating motor complex, which is what triggers peristalsis. It is a basic electrical rhythm that is found in all of the digestive organs, and this will occur until the next meal. The mechanism is by a hormone called motilin.